Good morning, everyone. This is Christy Sullivan. I am the Secretary of the American Society for Cellular and Computational Toxicology. I'd like to welcome you to today's webinar presented by ASCCT and the European Society for Toxicology in Vitro. If you're not familiar with our organizations, both of our societies host annual or biannual meetings and pursue other activities to promote computational and in vitro toxicology fields, and also have a focus on supporting early career scientists and offering training and education like this webinar today. I just wanted to briefly give you a couple of announcements. We have 2 webinars already scheduled for next month and the month after. You can see the titles here and the dates and times. They will be available for registration in about a week on the website. For right now, ASCCT and ESTIV members can register, and you should have received a link in your email. We also have some annual meetings coming up. Actually, the ASCCT meeting is coming up very soon, next week. There is still on-site registration possible. So if you are in the DC area and are interested in attending, please visit the website to find out more information. The ESTIV annual meeting is coming up next June, and you can visit their website there for more information about that meeting. If you are interested in showing a webinar or if you have suggestions for topics for webinars, we try to give one once a month. So just get in touch and we can talk about scheduling one, or we would be happy to take your suggestions. Just briefly, some housekeeping issues. The webinar is recorded. ASCCT and ESTIV members will have access to the recording shortly after the event. Everyone will receive a modified copy of the slides. And the lines are muted to cut down on background noise. After the presentation, there's going to be a question and answer session. And there are 2 ways that you can ask a question, and we welcome all your participation in the Q&A. The first way is to type a question into the question box. So you can see on the little GoToWebinar control panel, a question box. Go ahead and type your question in there anytime, and we'll ask it after the presentation. Or you can raise your hand and I can call on you, and you can ask your question that way. Today's presentation is by Lowry Curley. Dr. Curley is the CEO of AxoSim. Lowry received his PhD at Tulane University in biomedical engineering and completed postdoctoral research in Antwerp in collaboration with Janssen Pharmaceuticals. Subsequently, Lowry has served as the Director of Product Development and the Director of Research with several biotech startups. He founded AxoSim in 2014 to revolutionize the search for cures to the most devastating neurodegenerative diseases in the world. Lowry has presented at international conferences, is named on several patents, and has received more than $4 million in small business awards, including from the National Institutes of Health and National Science Foundation. AxoSim's nerve-on-a-chip and mini-brain platforms enable biopharma partners to deliver more effective and safer drugs with greater speed and efficiency. So we're looking forward today to hearing from Dr. Curley on the platform for assessing peripheral neuropathy. And with that, I'm going to go ahead and give you control.
Perfect. Is everyone able to see my screen?
Yes, I can see and can hear you, so please go ahead.
Fantastic. Yeah, so first, Christy, thank you for the opportunity to give this presentation. Certainly excited to share our science and our data with the broader field, and have always appreciated working with you. So to kind of give you background, you had a little intro to what we do, but particular to the nervous system, some of these alternative in vitro testing has lagged a bit, and that's despite the fact that there have been numerous publications that have shown that three-dimensional organization is particularly important for electrical activity of neuronal cultures. So this was a publication 7 years ago now that showed, if you see on the left, that structurally neurons in 2 dimensions, which you see at the top, are very different than a tissue explant of the superior cervical ganglia that you see at the bottom. So very long, spindly type connections in 2D. And then if you look in 3 dimensions, you can see the beginning of organization that is closer to the way that these neurons communicate with one another. Now, more importantly, perhaps, than the structure is the electrical function, because I think, everybody understands that that's the critical part of making sure everything works appropriately in all of our systems. So what you can see at the top right is staining for voltage-gated calcium channels, and you can see in 2D that their expression is very punctate. So it's sporadic, and then if you look at the 3D and the tissue explants, those are really a lot more similar. And then when you look down at the magnitude of responses to stimuli in 2D, 3D, in that intact tissue, you can see that 2D really is more hyperexcitable and hypersensitive. 3D gets you better responses, in predicting what would happen with intact tissue. And the result of this is that a lot of work in toxicology and, even in disease modeling has been skewed because of this hypersensitivity. And so a lot of times, we'll see toxicity maybe before there really is toxicity in animals, and it makes people kind of trust these systems a little bit less. So, kind of the next wave, you talk about 3D, and that's a critical junction, but then to bridge the final gap, something that's gaining a lot of traction are microphysiological systems. So these are organs on a chip, these are organoids, and, we're answering right now, are they more relevant than in vitro assays? And there's a lot of publications that are showing yes. What will take a bit longer is showing that they're more relevant than animal models. And a lot of that just is because, finding clinical results to compare that to takes time. Now, a lot of other tissue providers and microphysiological system developers are already starting to show this with historical compounds, and I'll show you a little bit of that data that we've used with our system. We have now 2 systems. So our nerve on a chip is technology that we licensed from Tulane University, and that really mimics the peripheral nervous system and some of the spinal system, where this long organization that you can see, this polarized structure, is critical. And I'll show you a bit more about what that looks like. And then there's some new technology that we license out of the laboratory of Dr. Thomas Hartung at Johns Hopkins. And, he's really a pioneer on this system and has done some great work. And something that ties these systems together and is very unique is that both of them have high levels of myelination. So, the more complexity and the more you can mimic the natural system, the idea is the better that the translation will be. So, the peripheral nerve models have really been something that's lagged behind, and there are unfortunately very few of them out there. And I think that's because the central nervous system, that has more pathologies, and any kind of disruption there manifests itself perhaps a bit more obviously. But in the peripheral nervous system, when you're talking about neuropathies, there are rare diseases, and these can be just as crippling. And so this is a system that we originally set out to mimic. Our Nerve on a Chip platform is the main platform I'll be discussing here. Obviously, that's in the title of my talk, but if there is time, I will also touch on the mini brain technology as well. So I mentioned this polarized structure that we have. You can see at the top that on the left we have neuroglial spheroids, and then we confine that into this fiber tract. And I'll actually show you a video so that you can get a little better idea of what that looks like. And the thing that's really nice about our system, these are untraditional cell culture inserts. So the way that you interact with these, the maintenance, the feeding, the assays is quite similar, and it makes it, I think, a bit easier for us to translate. So on this video, this truly is a platform, and there's a number of applications for this. So we can pick and choose the cell types we want to put into our system. One of our most common is combining Schwann cells with human iPSC motor neurons. We form these spheroids, and we're able to engineer this into our nerve on a chip. So that lets us isolate the neurons into this circular shape of the keyhole, and then define and control through chemical and mechanical cues the axonal outgrowth itself, as well as migration of Schwann cells. Oftentimes we'll use motor neurons and astrocytes. It really depends. In here, we have approximately 50 to 100,000 cells typically, and that means that we have this dense outgrowth that lets us get electrophysiological metrics out of this system that has previously been unavailable. So rather than going in and doing patch clamp, we're able to look at the entire population, and there's value in both, but looking at population level responses really better mimics clinical results. And this is truly something a neurologist would do if you had, let's say, a neuropathy. After we understand how a drug may affect this system, we're able to come in and look at the structure. And we can dive in here. We can look at, what's the density of nerve before and after treatment, or before and after a toxic insult. What's the size of these, and then diving into the ultrastructure. So here you see nice, compact, mature myelin, and seeing perturbations to the structure and correlating that with the functional outputs really lets us start diving into mechanism of action in a way that has previously been unavailable in vitro. And again, just to touch on the fact that these endpoints are clinically relevant, it makes this extremely unique. And what we have found is that builds in a comfort level with the data that we've generated, because it's a lot easier to extrapolate to what has been seen in vivo and clinically. You don't have to necessarily compare, apples to oranges when you have 2D data, and then you compare that to clinical results. The outputs just simply aren't the same. So we allow that gap to be bridged. Again, I mentioned already this myelination being a very unique part of our system, and a very important part, because myelination is really, what causes the function of your system to work the right way. And a lot of rare diseases have disruptions in this myelin, which means functionally, your signals get weaker as they're transmitted through your body, and sometimes they're even fully interrupted as this myelin degrades. So what we did here and published last year through Tulane University, who we maintained a partnership with, is we see that in about 4 weeks, this myelin, quote unquote, matures. So you can see the thickness and the tightness of that myelin wrapping increasing up until about 4 weeks. And we have taken this out further, but what we see is the system doesn't change much after that. So what you're looking at on the bottom graphs is the amplitude of the signal we record increases as this myelination increases, which again we would expect. And then the conduction velocity, because this myelination is supposed to speed that signal up, and we do see that happens. Now we have taken the system all the way up to 8 weeks as well, and I'll show you some reasons why that's important. But again, the myelin sort of plateaus at about 4 weeks. The main data that we've published so far has come from using this system with rat tissue. So, as we were getting started validating the system, this was much more accessible. And, we found that we can certainly reduce the reliance on animals by utilizing this system. But ultimately, the idea and some good data that we have is around humanizing the system. So what we have shown in that is extremely important. I mentioned we can go up to 8 weeks total. What we typically do, grow this to 4 weeks in healthy conditions, and then we will expose this in the situation you're seeing here to paclitaxel. Typically in vitro tests, cap out at about 72 hours. So that's sort of where we started our system. And we saw that we actually didn't see much of a response at 3 days. So what we said was, okay, let's take this system up to something that's more relevant, because 3-day exposure time, for chemotherapy is just, that's not how it's done clinically. So we pushed this to 5 days, and we started seeing a response. And as we took it to 7 days, we saw this more nuanced response that actually correlated better with what had been seen in vivo. So since then, we typically now do about 2 weeks of dosing, but we have in one situation taken dosing up to 4 weeks. So that's the 8-week total culturing. And we did this in collaboration with one of our pharma partners. And what we found is at 28 days, at about 3 weeks, we started seeing a toxic result. Didn't see anything before that. At 4 weeks, we really saw, a nice dose response curve. And once we've talked to and understood the whole background on this, what we found was the scientists at this pharma company had not been able to recreate anything in vitro that they saw in vivo. And the first time they saw in vivo responses was at 28 days. And we were actually able to show the exact same results. So this really shows you, one of the knocks on in vitro is the exposure time isn't in vivo or clinically relevant. And we're building on that to try to get towards better relevance. So we've done a lot of validation data. This is just a little snapshot. Primarily, we've worked with chemotherapy-induced peripheral neuropathy to date. And so you can see 4 of the main classes, of chemotherapeutics here. Some questions that we got early on that were really important ones, and we're about to publish on some of this data, is, people ask us, well, okay, you see this functional response, but what's, how do you know that's not just the cells dying? How do you know that those things aren't directly related? So we set out to explore just that. What you see in green is CCK8 assay. So that is a metabolic analog for cell viability. And we see with paclitaxel and vincristine, the functional decreases and the cell viability analog are decoupled. So we see functionality decreasing, before we see any significant loss in cell viability. And, when you think about mechanism, it makes sense. These are, microtubule, either stabilizers or inhibitors, and don't directly affect cell health. Or at least it is believed that way. Not everyone agrees on the toxic mechanisms of action of these, and there's obviously some gray areas. But when we look at oxaliplatin and bortezomib, where we would expect more cell viability issues, we do see that. Now with oxaliplatin, we see that they track pretty closely. And then when you look at bortezomib, they certainly mimic one another, but we do see that functionality being a little bit more sensitive. So then we're coming in and doing more looking at the structure of these. So, the functional testing is something that we can get quite quickly, but, looking at histology, it takes a lot more time. So this data is really coming in almost every week. But what we're seeing with a couple of these is by going in and looking at the density of nerves, we see with paclitaxel, and then we see with vincristine that these track with one another. And then when we come in and look at other responses here, for instance, density of myelin rings and then density of cell nuclei, we actually see that there begins to be some differentiation between the different drugs. So as we're building this data set, all this goes into looking at this toxic mechanism of action, and ultimately where we want to go is have enough correlation that we can test for functionality and be able to extrapolate that to structure, and then just dive into the structure when we really want to confirm particular features. So we've also looked at some of the histomorphometry of the distribution of axon diameter. So blue in this case is a control, and this was in conjunction with the pharma partner, so we've de-identified this, but you can see with some of the compounds, you see sort of a flattening out of that distribution. And this is actually to be expected with some of these drugs. We can also, with electron microscopy, go in, look at G ratio, how thick is that myelin, how does that compare to axon diameters. So there's a lot of metrics that we can pull out of this really high content system. I mentioned, our ultimate goal is humanizing this system. So a lot of the work we have done has been with motor neuron cells from cellular dynamics. And we're able to get great growth with these. We're able to get growth that's just as dense as we have with tissue explant. And we've done this both with a monoculture model, so motor nerves alone. And then we have also done this with co-cultures. So we do it with motor nerves and Schwann cells, which is shown here at the top. And then we have also done astrocytes and motor neurons as well. We actually recently received a grant to work on an ALS model. So we are also able to work with patient-derived cells in this system. And as we're building out more applications for this, we're working on adding things like microglia, really increasing the complexity of the system. But of course, we have to understand each and every step along the way. And this myelination data is something that we just published, and there's a lot more information in there than I necessarily shared here. But we have seen, again, physiologically relevant myelin in our human-on-human system. And as far as we are aware, this is some of, if not the first publication that shows this really compact, mature human-on-human myelination. A lot of people have done combinations of either rat Schwann cells and human neurons or vice versa, but we were able to do this with a fully human system. In this case, I'm only showing a snapshot of the validation data we have done. Again, this is using historical compounds that have been tested thoroughly in vitro, been tested thoroughly in vivo, and then also clinically. And so this is an example of looking at bortezomib. And what we're doing with our human system is comparing our 3D functional outputs with traditional 2D metrics. So in this case, what you're looking at on the left is our nerve conduction velocity response to 2-week exposure to bortezomib in this case. And we are on the right looking at 2D high content imaging. So looking at structural imaging with 2-dimensional cultures. And you can see here they actually track pretty nicely when you're looking at some of the different outputs of the image-based analysis. But we have found, very interestingly, examples where that isn't necessarily the case. So paclitaxel is one that we looked at, and in the range of dosing that we tested, we do see a dose response with our human system on the left. But then on the right, going up to 100 nanomolar in this case, you see perhaps a trend starting. So we are going back and we're going to bring this up to 1,000 nanomolar and see what we see. But if you had stopped your dosing at 100 nanomolar, you potentially could have missed this in 2 dimensions. Now, even more powerfully, we worked with a substance called teriflunomide, and here we saw a nice, pretty linear dose response with the functional conduction velocity testing. But then on the right, at the same dosing range, did not see a response at all. So this is a drug that could have slipped through your in vitro testing, and ultimately was actually not seen in vivo to the extent that it ultimately popped up clinically. So this was a drug that perhaps should not have gotten to clinical trials, but ultimately did. And so, this is kind of the first example, and there's more that we're generating to showing that, our system is necessary, and our system really is predicting human results and doing that from the benchtop. So we do have time for our mini brain platform, which is exciting. So all of these publications have been from the Heart-Tongue Lab at Johns Hopkins. And back in 2016, they were able to characterize a system of these three-dimensional spheroids that are directly differentiated from iPSC. And what they were able to find, which we're recreating in our facilities right now, is that you get a really nice, diverse neuronal cell population. And then you're able to get dopaminergic, glutamatergic, GABAergic, and some other more specific subtypes of neurons in that system, as well as astrocytes. And then perhaps most uniquely, and what is the most exciting part of this, is that we get markers for oligodendrocytes here. And this is one of only a few systems that I'm aware of that actually gets oligodendrocyte differentiation and then ultimately myelination. So this is work that, again, has been published by the Heart-Tongue Lab, where they are able to see co-localization of neurofilament and MBP staining. And you can even go in and do electron microscopy on these, and you can see both mature and immature myelin. And in the case of on the right, with 8 weeks of differentiation, you see this nice compact wrapping that, again, very, very unique for an in vitro system. So they characterize this, and again, it takes about 8 weeks for the system to mature, and they get about 40% levels of myelination in that time course. So they have gone in and also looked at demyelinating compounds. So in the case of what you're seeing here, cuprazone, which is something that is used in vivo to test for things like multiple sclerosis and some toxicities, as well as developmental toxicities. And you can see here that you do see a dose-related decrease in myelination. So this is one of the first in vitro assays that has that level of myelination, and it lets you look at some of these structural components that are really, indicative of a better level of model, which suggests that it will have a more predictive and more detailed set of data that you can capture from this system. This has also been looked at in a developmental neurotox study, so in this case with rotenone. And what was seen here that, toxicity wasn't necessarily just seen across the board for neurons. In this case, as expected, it was dopaminergic neurons that responded to this exposure. So you can see here that, this lets you dive in, not just on, is this toxic? Are there pathologies that are being manifested as a consequence of exposure to drugs, but what's the potential mechanism of this, the why, not just the fact that it happens. Neuroinflammation is another set of work that's been done by the Heart-Tongue Lab. As I mentioned, that's something we're also building into our nerve on a chip, becoming more and more of a hot topic. It's being implicated in almost every neurodegenerative disease that is unfortunately affecting almost everyone across the world. And so these are models that really need to be built. So in this case, the microglia incorporated to the mini brains after the mini brains were differentiated. And they were able to see with exposure to the flavivirus that if you look at the top right, on the left are the mini brains or brain spheres alone. So you see that there's really no significant results to that exposure. You look on the right, that is microglia alone, and you can see the only markers are TNF and CCL2. But when you combine the two, you see expression of response more indicative of what you would expect to see. So the markers IL-6, IL-10, and IL-1 beta all do show response only when the system is together. So again, reinforcing that you need additional building blocks in these systems to get responses more indicative of what one would expect to see in an entire system. So I went through that quickly, so I think that hopefully means we have plenty of time for questions. But I hope what you've been able to see is that this nerve-on-a-chip delivers some really unique metrics and results. And what that enables us to do is better predict how these drugs will ultimately work in the clinic. Now, there's obviously a lot more work to be done to fully, get adoption of these models, but we're certainly not the only ones working on it. There's publications coming out seemingly every month demonstrating a lot of this. And then, this is also, being built extensively within the central nervous system. So, our ultimate mission is really to be able to address any and every neurological pathology or issue that may be seen and be able to better predict, generate better data, and hopefully make a better impact on, being able to develop therapeutics to address a lot of these concerns and pathologies. So with that, I will finish and certainly take any questions anyone has. And if anyone would like to get in contact with me, I'll have my address here, so please feel free to reach out.
Well, thank you so much, Dr. Curley. That was a really nice sort of introduction and some data on the models that your company is making. I was wondering, first of all, and again, just to remind people, we have a couple of questions coming in. Just enter your questions in the question box, or you can raise your hand. I was wondering if you could say a little bit about, or expand on what you said at the end there about application. Have you had any discussions with either companies or regulatory agencies about using any of your models for submission, for regulatory submission? And what do you think is needed for that to happen?
That is an excellent question, and one that I think almost every stakeholder in drug development and regulatory agencies wants, is working towards, certainly. So, what I will say, and where this, has to start is, with usage by kind of the end users, and those are the pharmaceutical companies. Mm-hmm. What we have seen is a very interesting shift and maturity of a lot of the work that's going on here. So I'll try to touch on kind of each of the aspects of that you mentioned. The first being, again, adoption by these pharmaceutical companies. But truly, even before that, before I get ahead of myself, it's, where has the funding for a lot of this come? So, the primary drivers of this so far, initial funding was a collaboration largely between the DoD, and that was in combination with the NIH, and that's what funded a lot of this academically at at places like Harvard and a lot of other big institutions. And there was significant funding that came out of that, because there was some interest, and then there was enough data generated there that momentum continued to carry out, largely driven by the National Institute of Health. So they initially funded a lot of this for neurotoxicity. I think that was one of the first areas that people saw there being a need, and from a lot of this academic funding came some commercial entities that arose from that. Mm-hmm. And the NIH, I think, recognized that it's going to take commercial entities to drive this in a way that has expansion potentially across applications. And so, the NIH has really been pushing on that, and they are moving this, from tox now with a much more of a focus on disease modeling. And I think disease modeling is where, the potential can explode. And along the way, I know that the FDA and Dr. Fitzpatrick over there has kind of gotten a lot of interest with this. They've worked with a few commercial partners on, what could this look like for adoption. But I think those are still a little bit in the early days, because obviously there's a lot of, regulatory components that need to be hashed out there. But there is definitely interest. And I do know anecdotally of another organ on a chip company that has worked with a pharmaceutical company, and that pharmaceutical company has actually taken their data to the FDA when they were discussing their IND package. And so that I think is where things really happen. We have to work in concert with the FDA. There's a lot of proactive, talk there, and a lot of proactive activity as well. But once a pharma company sees, hey, this data is better, more predictive, it's faster, they have the resources to really drive this. And so, that's where we're trying to work. We have had adoption with quite a few pharma companies, and it followed the same trend of shifting, from neurotox. And once we got a good set of data in neurotox to basically, convince these people that what we were doing was valid, we now have some of our partners passing that information to their CNS and PNS disease modeling groups. And so as a result of that, they're saying, all right, well, you proved this in neurotox. We have absolutely nothing that we fully trust in disease modeling. So we're actually going to pay you to develop, systems, adaptations of your platform that will allow us to address some of these answers for MS, ALS. So I think as that adoption shifts, everything's going to move a lot faster.
Mm-hmm. It's interesting. We've got a couple questions. The first one is, how reproducible is the data that you've shown? So, for example, from one nerve on a chip to the next, have you been able to document that for the same compound you get a reproducible result?
Yeah, we have, and that's data that I took out of this that I should frankly add back in. We have seen that there is variability. Unfortunately, a lot of that variability seems to be coming from the cells themselves that we add in there. Especially with the iPSC cells, that technology is still evolving. And so we have done a lot of that. There are acceptable levels of reproducibility in there. But what we've found that works best to use this system is actually to spread out our testing across 3 different batches or 3 different weeks. So what that means is we'll typically do an N of 6 in our system. And we will do 2 of those samples in, let's say, week 1. We'll do 2 more samples in week 2 and 2 more samples in week 3. And when we've looked at the difference between 6 in 1 week versus spreading it out, we actually see that the reproducibility is a lot better, because it does account for that, within each drug versus testing a drug, from one N of 6 to another N of 6 a few weeks later.
That's really interesting. I know that these models built on human cells are now having to deal with human variability, which is kind of a new blessing and a curse at the same time, perhaps.
Yeah, I mean, it's an interesting topic, and one that I think at first we were a bit frustrated, with is we would either get cells or, whatever the case may be, and then even within the same lot, there were some differences. And so we've really tried to account for that through a stringent quality control system. But for neuro applications, the differentiation and getting the right expression of all the receptors, it's just difficult. And so what we found is, accounting for that in the best way that we can. And then we're also starting to work on, patient-derived cells, I think a little bit of what you were alluding to just now. And there you add an entirely different variable to the system. And I think that's something that we're all, gonna work to address. One of the biggest challenges that we're facing right now, and again, I think this is kind of echoed across the industry, is throughput. So we're able to use, these systems to test 6 to 10 drugs, let's say, and then deliver data on that, and that's helpful. But ultimately, we need to move back to where we can do, 50, 100 drugs. And there, that kind of a throughput will also let us test, multiple patient cells so that we can start accounting for more of that variability. But as of now, these systems are not high throughput. It's hard to get above 96 wells. And some systems, if you want to build in a lot of complexity, we're working even towards that. So there are challenges, but we've made a ton of progress, and frankly, the challenges are kind of fun to work through.
Mm-hmm. Another question, is there any difference between mini brains and NPC neurospheres?
Yes, there certainly are differences. There's a few different model systems out there, I think, of brain spheroids, however you want to call them. Part of it is in the way that they're differentiated, which, as I mentioned before, these systems are very sensitive. So you take it down, one pathway, you're going to get different cell populations, different phenotypes of individual cells. And so the mini brains, what's nice about that is there's a large level of control over the reproducibility, and that starts with the size of the brain spheres. And this is a big differentiator. Is one of the things that's concerning as you move into more three-dimensionality is diffusion. So is there going to be a necrotic core within this organoid? And what Johns Hopkins found and has shown is that if you keep these to about 350 microns, that makes sure that negates any of the diffusion issues to limit that necrotic core. So that's kind of a big differentiator, and being able to do that reproducibility. But then again, I think the biggest one is the myelination. So figuring out how to differentiate this with that complexity. But there's a lot of systems out there, and I think, for different uses, a lot of those systems, are very, very important. The mini brains are a bit of a, generic, for lack of a better word, cortical representation. But if you want to get better focus on differentiation towards, midbrain neurons, whatever the case may be, that takes an entirely different model. So, we need all of these in concert, I think, to answer the questions that are so important.
Mm-hmm. Great. Well, that's all the questions we have. So thanks for taking the time. I don't see any last minute hand raises. Nope. So I think we'll end the webinar. Dr. Curley, thank you so much for your presentation. And thanks to all the attendees for listening, and looking forward to seeing you at our meeting or at one of our next webinars. Have a great day, everyone.
Thank you very much.
Thank you. Bye-bye.